let's come towards the next question, that is question number 10. In the medium where E. coli was growing, lactose was added, which induced the lac operon. Then why does lac operon shut down sometimes after addition of lactose in the medium? So we know that lac operon is active in the presence of lactose. Lac operon is active in the presence of lactose, and whenever the lactose is absent, the repressor binds to operator and do not let the genes to be transcribed. Now, this lactose when present, it binds to the repressor and it codes for some enzymes such as beta glycosidase, permease and trans-acetylase. The enzyme beta glycosidase converts the lactose into glucose and galactose, which was readily available and readily used by the cells. Now, whenever the lactose is present or lactose is added, the lactose once gets used, the lactose is completely used, the lac operon shuts down, because in the absence of lactose, the repressor binds to operator and do not let the genes to be transcribed. The permease enzyme, which was encoded by the lac operon, enhances the permeability of cell membrane so that lactose is transferred into the cell once it is available. So, low level expression of lac operon is always required. Now, let us write the answer. Answer number 10. We know that lac operon is active in the presence of lactose, in the absence of lactose, the lac operon do not let the genes to be transcribed. But whenever the lactose is present, it will bind to the repressor. So, in the presence of lactose, it binds to the repressor. And encodes the enzymes such as beta glycosidase, permease and trans-acetylase. So, whenever the lac operon is added, after complete usage of lactose, the repressor shuts down and do not code for anything, because repressor in the absence of lactose will bind to the operator and this does not lead to the formation of these enzymes. Now, this beta glycosidase, what is the function of beta glycosidase? It converts the lactose. Whenever the lactose is present, the lactose will bind to repressor and forms these enzymes. After the formation of these enzymes such as beta glycosidase and permease and transacetylase, these beta glycosidase converts the lactose into glucose and galactose. Glucose and galactose, which is readily used by the cell, and the permease enzyme enhances the permeability of cell membrane. This is the function of enzyme permease. So it enhances the permeability of cell membrane so that the lactose is transferred. Into the cell once it is available. Once the lactose is available, it can easily transfer into the cell because of the increased permeability of cell membrane with the help of this permease enzyme. So low-level expression. So always low-level expression of lac operon is always required. Why it is always required that lac operon should expresses and forms these enzymes? Because after formation of these enzymes, permease enzyme will enhance the permeability of cell membrane so that once the lactose is available, it can easily transfer into the cell. So this is the complete solution of question number 10.